Nature's Archive Podcast, a Jumpstart Nature production.
If you've listened to more than a few of my episodes, you've probably noticed some common themes. One in particular is that everything in nature is interconnected in multiple ways, and today's episode demonstrates that in some mind-bending ways. What's the topic? Well, it's an often overlooked subject, and if it's handled well, it will sequester carbon, reduce the impacts of droughts, improve your water quality, and probably save you money. And if you need another hint, when it's healthy, it makes your food more nutritious. So by now you probably guessed the topic. It's soil ecology and the soil food web, and my guest is none other than Dr. Elaine Ingham. Dr. Ingham has over 4 decades of experience in soil biology and is generally recognized as the foremost expert in the field. She's the primary author of the USDA Soil Biology Primer and the founder of soilfoodweb, Inc., a group dedicated to empowering ordinary people to bring healthy soils back to life. Our wide-ranging conversation only scratched the surface of soil ecology. As I said a moment ago, everything is interconnected, so it makes conversations like this somewhat challenging. Like soil ecology itself, everything interrelates. So what did we cover? We talked about the difference between dirt and soil is basically an ecosystem unto itself, full of fungi, bacteria, predators, parasites, and more. Dr. Elaine discussed the entire process of how fungi, bacteria, and other organisms convert soil nutrients to forms readily available to plants. It's really fascinating. And she discusses how plants trade sugars for nutrients from the fungi. We dig deep into how to measure soil health, and that includes how to compost and how to assess both soil and compost with a microscope. It's not as hard as it sounds. And in the show notes, we have a couple of videos to help with that. We touched on many other topics, too many to say here in the intro. I would spoil the whole show. So without further ado, Dr. Elaine Ingham. Dr. Elaine, thank you for joining me today.
Glad to be here.
You were actually requested by one of my listeners to join the podcast. I think I neglected to tell you that before. So I'm really happy that I'm finally able to cover this important topic.
Great. Yeah, the fandom is out there.
Yeah. So in general, I think that soil microbiology and soil health is one of the most underrepresented and most important aspects of our general environmental crises that we face today. And I'm hoping that we can get into many aspects of that today. And before we jump into that, I would like to learn a little bit about you. Can you tell me, how did you really first get interested in nature, and how did that then translate to soil microbiology?
Well, yeah, it really comes from both of my parents. Really liked to be in the out of doors. We would go hiking. We would go on picnics. We would go and spend a weekend up in northern Minnesota. I remember a couple times where, it was all hot, late September kind of temperatures, and went up to northern Minnesota to hang out for the weekend in the forest, do some hikes and do some swimming in the lake. And the first afternoon we were there was a perfect summer day, just wonderful. And we all went swimming in the lake, and we, got back into the tents, and we didn't want our sleeping bags on because it was so hot. And sometime during the night, we all started pulling our sleeping bags back up over us and just started to freeze to death. By the time we got up, there was 3 inches of snow on the ground. And, we were always going out doing that. Every year when I could be trusted to be on my own, my friends and I would go up to northern Minnesota and canoe into the Boundary Waters and up into the Quetico. Had some grand times. I was always 2 or 3 weeks out of doors canoeing someplace and fishing for meals and things like that. My father was something of an Indiana Jones character. We went out to various places overseas even, and always interested in the wildlife, the organisms that we're seeing. And I showed a definite predilection for understanding the microorganisms in the water and in the soil. The first microbiology class I took at college, I knew what I wanted to do for the rest of my life was to work on those microorganisms. When I started my PhD, though, it was really interesting that my major professor told me about the project that he wanted me to get involved in. And it was all about fungi and how you measure active fungi, how do you determine total fungi, getting into some of those fairly basic questions. He told me I had to go and speak to professors that were in the soil science, growing plants, horticulturalists, agronomists, the soil agronomy department, and go and talk to all of them. And I remember every single one of them. I would go into their office, start talking about my project, and this look would come over their face of complete and total boredom, because and I would finally ask, so what do you think of my project? Do you think it's good enough for a PhD? And they said, well, technically it's good enough for a PhD, but you shouldn't do that work because you have to have a job lined up once you finish your PhD. And there's not anybody going to hire you because the microorganisms in the soil, they're just there. They don't do anything. They don't help plants in any way, shape, or form. They just, they come back after a disturbance and they're just there. And that's depressing when your heart is set on doing these sorts of things and looking at and understanding biology in the soil. But at that time, the culture was not to be that domestic or worrying about the house and what Mother Nature meant for things to be happening in the soil. And I did have to take it back a bit in my life why was I so interested in these microorganisms in the soil? And it was because we didn't really understand what it was they did. And nature doesn't have something hang around for 4 billion years if there's not some use for them. They're doing something. It's got to be important or they wouldn't still be hanging out. So it was an uphill battle to convince people of why soil biology was so important. And it's very satisfying to me to be here 45 years later and saying, finally, everybody is understanding how important biology in the soil is. We would not be here without those microorganisms in the soil. And we had better stop destroying them. If we want humans to survive, these organisms have to survive. So we got to get away from the pesticides and the inorganic fertilizers and the constant tillage and herbicides. Got to stop that, turn it around and get that carbon back in the soil where it belongs.
Well, I think your story is really instructive in a couple of different ways. One, sticking with something that there's something there, but then also the lack of sort of first principles thinking that was demonstrated by those advisors that you had, that you articulated really well. The fact that, well, why would these things be there after all this time if they weren't doing something useful, which I think we need to remind ourselves of, like first principle thinking.
Yep. That even though professors that should be knowledgeable about soil didn't have the least clue of the most important thing. Back in the 1970s, 1980s, they just turned a cold shoulder because, well, where did all their money come from? It wasn't from the microorganisms. It was from all those chemical companies that made billions of dollars a year selling people materials that would kill, specifically destroy that biology in the soil. And they didn't want people to understand that's what they were doing, was destroying our soil and turning it into dirt.
So actually, that's probably a really good lead in to what is healthy soil? Like you talked about soil turning into dirt. What's the difference?
Well, when we're looking at dirt, we're just looking at the sand, silt, clay, rocks, pebbles, parent material, boulders, those sorts of things. So when you've completely destroyed the biology that's supposed to be there, now you're dealing with dirt, and good luck growing plants in dirt. So what is good soil? Well, you have to have the mineral component, the sand, the silt and clay, the rocks and pebbles, those sorts of things. But you also have to have organic matter in that soil. And you have to have at least 3% organic matter, or you're not going to get the benefit out of that organic matter being present. Okay, so why organic matter? And why organic matter that is as diverse as you possibly can make it for your bioregion? Because that's food to grow the microorganisms. We have to have all of those organisms in the soil because we have to have oxygen moving down deeper and deeper into the soil So that means your soil, all those sands, silts, clays, rocks, pebbles, leaves, twigs, all that organic matter has to be built into some structure. You take the individual particles and you start gluing them together. The bacteria make copious amounts of glue, lots of different kinds of glues in that soil, so that they can build the micro aggregates and make, well, I like to think of them as like individual houses. And then if you're in a neighborhood that has lots and lots of bacteria, you better have apartment buildings. And how tall is your apartment building going to get? So some of those micro aggregates get pretty much on the end of macro aggregates. And so to form the macro aggregates, so you have even more space between those micro aggregates. Let's pull some of them together. So that's fungal hyphae. With their strands, they'll pull this aggregate in, this micro aggregate, this other one in, and now we have aggregates that we can see with our eyes. And it is fungi that are most important for building that level of structure. More space to allow oxygen and water and your roots to grow as deep down into the soil as necessary. If you want to not worry about your property burning when there's a wildfire going through, if you want to not have to apply massive amounts of water late in the growing season when it gets very dry down, you need to have that structure, and you need to have held all the extra water that came to your property at snowmelt in the wintertime or as spring rains. You want to have all that extra water filling or half filling all of the pores deeper and deeper down into the soil. When you look at most large conifer trees or large deciduous trees, their root systems go 100 feet, 150 feet, 250 feet down into the ground. And they're after that water in the late growing season so that they don't die. Well, if you've killed the biology in your soil, you won't have any of that structure. There'll be lots of erosion. You'll be losing your soil out into the rivers and lakes and streams. You will not be able to hold on to nutrients. So there's a lot of leaching of the soluble nutrients. We need these organisms. They do so many important things. I've only touched on a couple of them so far. I have a suspicion we'll get to them all before we get done with this podcast.
Yeah, I think that's a good start. And it's a good visualization too, where the roots of these plants wouldn't really be able to get deeper in the first place if the soil's so compacted, if you didn't have the fungi helping. So there's two benefits provided simultaneously: adding the space in the soil for the water to penetrate, and then also allowing the roots to go so deep. In addition to what you just talked about, my understanding is that there's a lot of symbiosis. There are different organisms working together in good soil. For example, bacteria that's helping to unlock the nutrients within the soil. Is that accurate? And can you tell me a little bit about how that works?
Yeah, sure. There's a lot of synergism going on in soil. It's more the standard operating mode than it is to have the little things get eaten by the bigger things get eaten by the bigger. And it's important in soil, but it's much more important to be going through the symbiotic interaction. So there's actually probably three things I want to cover in this making nutrients available to your plant section, because that's such an important thing. And it's, because we've destroyed that biology in the soil, that's why you have to use inorganic fertilizers. That's why you have to use pesticides and herbicides. So it's really critical that you get this part of the food web up and going as rapidly as you can. So when we're looking at a plant starting to grow, the seed pops open and the roots start moving down into the ground. Most of the nutrients present in that seed are actually going to be going to the purpose of making exudates from those roots that are starting to grow into the ground. You have to have enough energy and nutrients for that seedling to put its leaves above ground and start photosynthesizing. That's really critical to increase that flow into the exudates coming out of the plant. The exudates are messengers. I think of it as the pizza delivery system in the soil that you call up the pizza delivery guy when you want to get some food. And so the plant is putting out this message in the form of an exudate, which is mostly simple sugars, proteins, and carbohydrates, but very specific to the nutrients that the plant requires. Whatever the plant is lacking, it's going to be sending out the message to the bacteria and the fungi in the soil about how much of what kind of nutrient the plant requires. And so here's this bacterium over here receiving the message, make this kind of enzyme so that you will pull this kind of nutrient out of the silica bilayer of the sand, the silt, the clays, rocks, pebbles, etc. And so that bacterium is taking up those nutrients delivered to the bacterium from the enzymes working on these substrates in soil. The fungi, of course, over here, getting a different set of instructions from the plant. So it's going after a different nutrient. And it sends out hyphae, and at the ends of the hyphae, releasing the enzymes that are needed to pull those nutrients out of the organic matter or the silica bilayers in the sand, silt, and clay. So the fungus is accumulating nutrients for the plant, is like the plant's pantry. The bacteria are pulling in nutrients, storing them in their biomass. It's the pantry for your root. And when there's a high enough concentration, then the predators of those bacteria and fungi go, oh, look at those great things to eat over there. Let us go over, chow down. Because the nutrient concentration in bacteria or fungi is much, much higher than what we see in the bodies of the predators of the bacteria and fungi. So they don't need near what's stored in the bacteria and fungi. So all that extra, all the excess nutrient is going to be released into the soil in a plant available form. Back when those nutrients were in the sand, so clays, rocks, pebbles, and organic matter, your plant couldn't access them, not capable of taking up that form of that nutrient. Not until the predator organisms release those nutrients into the soil are those nutrients actually something that the plant can take up into its root system.
Hey, Michael here. I'm sorry to interrupt, but nature needs your help, and so do I. Jumpstart Nature, my education and conservation organization, is at a bit of a crossroads. We're still awaiting the IRS's review of our nonprofit application, and since we filled out the long form, it means it takes several months for that to happen. So we're in limbo for another few months. While we wait, we still need your financial support, perhaps more than ever, because we're taking on additional expenses to operate while we wait for the official designation. So the easiest way to support us is by becoming a patron on Patreon, and you can do that by going to jumpstartnature.com/Patreon, spelled Patreon. You can join Sarah, Mitchell, William, Erica, Jess, Maria, and Lisa, some of our newest patrons, over the last few months. And you can do this for as little as $4 a month and cancel anytime. And along the way, you'll get a few perks as well. If Patreon isn't your thing, we also offer merch and a direct donate option too. Just check out the website. I hope that you can help, and thanks for being part of our movement. So to make sure I understand that correctly, the bacteria and the fungi can't deliver those nutrients directly to the plant. You require this extra layer in the system.
Yep. And it's hard for people sometimes to envision what's actually going on. And it's really important for plants to be getting all the nutrients they need in the proper balances. When you put out an inorganic fertilizer, you're only putting out one type of nutrient. And so your plant is going to be unbalanced. It's going to have way too much nitrogen, but not enough cadmium or zinc or boron or any of those other nutrients. So the plant still has to carry on this interaction with the bacteria and the fungi and the protozoa and nematodes. Still has to be carrying that on. The inorganic fertilizers that we use are all salts. They've got to have the positive and negative portions binding with something. And when that inorganic fertilizer gets into the soil, the concentration of the salt in that, which is the structure of the fertilizer, is going to kill a lot of different species of bacteria and fungi and protozoa and nematodes and microarthropods and earthworms, and lots of things die because of that. And so here we are again, preventing this normal nutrient cycling process from being able to happen. The more inorganic fertilizers you put on, the more you kill this biology in the soil, and you become totally dependent on very expensive inputs into your system. Whereas if you just make compost from your own organic waste, you get rid of another problem by making that compost. Then you can put that compost out, and you can put that highly diverse set of microorganisms that you need in your soil. You can be replacing them anytime you need to be replacing them. Hopefully you understand pretty quickly that you don't want to be killing these organisms off anymore. So you don't have to keep putting more and more compost out. Start protecting your beds, start protecting your fields so that you don't have to put these organisms out. There are a lot of clients of ours that have brought the biology in their soil up to what the plant requires, and they're not putting compost back out on the property anymore. It's not necessary. All the organisms are in place. How do you know that the organisms are all out there and in place? You've got a microscope. And so you're going to be using your microscope to look at what's actually in your soil. And if the balances are right for the plant that you want to grow, you don't have to do anything. Now, if the things aren't balanced more or less properly, then you might want to go out and put on a gentle spray of a compost extract. It's very easy to apply, doesn't take that much compost. Or a compost tea you might need to put out in the first year or two if you have a lot of above ground disease causing organisms attacking your plants. Putting out a high concentration of these organisms will protect the leaf surfaces, top and bottom, so that no disease causing organism can even make it anywhere close to the tissue of your plants. So we get rid of the foliar diseases, the foliar insect pests. They don't touch your plants. They fly on by. They don't recognize that your plants are plants because they don't have the smell, the gases that normally come off. The vegetation are no longer being released. They're being used by these microorganisms to make more microorganisms, protecting your leaves even better.
So there's a handful of things I want to follow up on in what you just said. Maybe I'll start with the last topic and work my way backwards. So applying these microorganisms to your plants on the leaves, can you tell me a little bit more about what that looks like? You mentioned compost tea. Is this sort of like a liquefied form of compost? Or
Yep.
How does one create such a thing?
So you make your compost, you take a sample, make sure that it's got the right balance for your plant. And now all you're going to do is, like in a container, depending on how much you need. If you just have a couple of plants in a windowsill, or if you have a small garden with 1 or 2 rows, you don't need much. You take a 5-gallon bucket and you put the compost into something where the water is going to flow through the fabric. So your compost is inside, all the big chunks stay in the bag, where all of the small bacteria, the fungi, the protozoa, the nematodes will be extracted. And you're just gentle and careful of that you're extracting all of the organisms off the surface of that compost. It does take about 80 psi to pull most of those organisms off. So you want to push your hands together, and the water rushing out will pull those organisms off the surfaces and into the water. So you do that, for maybe 3 minutes. No need to go more than that. Some people like to leave the bag hanging in the water. I usually like to take the bag out because we're going to then add a little bit of food to get those organisms active and growing in the ambient conditions. We want to have those organisms making glues that will instantaneously have them adhere to the surface of the leaf And then think about the fact that we've got to get the drops on the top of the leaf, but we also have to get those organisms to stick to the bottom of the leaf. So you have to have immediate sticking. The second that drop of water that you spray on that leaf touches the surface of the plant, we want all those bacteria and fungi and protozoa and nematodes to stick to that surface and start forming a layer where any of the disease-causing spores flying through the air lands on your leaf. There is a huge gap between where the spore arrived and where the leaf actually is. So we're going to allow the organisms that you've extracted from your compost. And people usually ask me at this point, what do you do with the leftover compost in the bag? You dump it on your compost pile. It adds a little bit of extra moisture, but that's about it. All those organisms that you ripped off, they're going to regrow on your material. It's just more food coming back to the compost pile. So during the next 24 hours, you want to be aerating what's in that 5-gallon bucket so that you keep it aerobic. You don't want anything to go anaerobic because the good guys grow in aerobic conditions. The bad guys grow in anaerobic conditions. There's a few minor exceptions to that rule. So you let that incubate, that bubble around for 24 hours, and then you turn off the aeration, put your compost tea into your sprayer unit. If you've gotten too much chunky stuff coming out through the bag that you were extracted, you let a lot of that material sink to the bottom and you take off just the liquid part because you don't want to plug your sprayer. And then you spray just as you would for anything else. If you have used your sprayer for toxic chemicals, those toxic chemicals have gotten into the surfaces and you would have to wash the surfaces quite a bit, 5 or maybe even 10 sprays for a minute or two to try to get all of the inorganic fertilizer or pesticides, toxic chemicals, to get that all washed off those inside surfaces. So a little bit of hydrogen peroxide or bleach going through the first couple washes, and then 6, 8 flushes after that are necessary to get all of that pesticide off the inside layers. So a lot of people will buy a sprayer just for their compost tea, compost extracts. So now with the compost tea, you go and spray your trees, shrubs, above ground parts of your tomato plants, whatever you've got, just making sure that you're trying to spray the top leaves of the leaves, and then you're directing that spray from below. Go ahead and spray your flowers, spray the fruit. You want to protect all of those surfaces from the diseases, and all of the organisms that are in your compost teas are organisms that will be beneficial to you as long as you maintain aerobic conditions and high diversity.
It's fascinating to me that the same organisms that are flourishing in the compost and in the soil ultimately can be applied to the surface. It's such a different environment, direct sun and everything else that's going on. But it's the same organisms, apparently.
There's a little bit of selection. The things that are really soil dwellers and they don't like sunlight, and there are no foods for them sometimes coming out of the leaves. But all of the leaves, the twigs, the bark, the flowers, the fruit, all of those release their own exudates. A great deal of that is meant to grow these organisms coming from the soil, because where did the organisms on the above ground part of your plant come from? Didn't come from the seed. It comes from all these cute little critters crawling around in the ground, getting their legs and their body fully covered with all these really good organisms, and they walk up your plant. And as they're walking, they're leaving behind inocula that the plant then feeds to get that protective layer all over them. So it's all starting in the soil. So we're speeding the process up by making the compost, by applying the compost as a spray. You're getting that layer put out there. It's also a good way to determine whether your neighbors have been spraying pesticides, because if they have sprayed pesticides and you happen to be unfortunately downwind, All you have to do is take some of your leaf material off and look at those leaves and determine whether you've got the organisms present on those leaf surfaces or whether they've all been killed by the neighbor's sprays. So what do? You want to then make up another batch of compost tea and spray your trees again, or shrubs or veggies or whatever you think may have been harmed by those toxic chemicals.
And when you say look at the leaves, is that again under a microscope?
Yep, using a microscope. And we don't need real high power. A lot of people have been told you have to have 1,000 magnification in order to be able to see. No, not true. And you can prove it to yourself. You start out with a 4x objective and you see all these little dotty things zooming around. Some of them are not modal, so they stay in place. So you go up to your 10x objective, take a look, and everything's just larger. Go to your 40x objective, everything's larger. And you're going to be saying, those look exactly like bacteria. They're little round guys, or they're short little rods, maybe longer rods, but you're looking at all the bacteria. So just by the 4x, 10x, and 40x objectives, 10x eyepieces, and you're good to go. You'll be able to see all the organisms in the soil that you want.
So we could talk a little bit more about using the microscope perhaps for a moment, since we're already partway down that path. And I've heard you speak before, and I know that there's a method by which you can assess the health of your soil. If somebody wants to see with a microscope how their soil is doing, what would they do?
Yep. Well, you take like a spatula, or you take spoons. A spoon works just as well and is a tad less expensive. So you can take a spoon and you dig down to the depth you want to look at what's going on. So maybe you want to look at your 0 to 5 centimeters or 3-inch level, and you just take a couple spoonfuls out of that, put that in the plastic bag, go to another place in the same plant, same treatment, that take another couple of spoonfuls and put that in the plastic bag, maybe go to a third place. So you're getting some idea of the variability within your system. You're making it more uniform by taking all these little bits from here and there. You then take your bag back inside. Don't start breaking up the clods. Don't start destroying the structure right away. You may be taking in several samples, so you don't start breaking them up until just before you're going to start preparing that sample for the microscope. So you bring your baggie inside. Now you mix it up so you make it as uniform as possible. Now you're going to measure out 1 gram or 1 milliliter volume. They both should come out about the same. So measure out your soil sample, 1 gram in a test tube. And now you add 4 mils of water. Please make sure that it is water that doesn't have chlorine or chloramine in it. So that means if you're taking water from your kitchen faucet or your bathroom faucet, you're going to have to neutralize the toxic chemicals that are in that water. You're going to have a container where you fill up a gallon of that water and put 1 drop, maybe 2, of humic acid, because that will complex all of the chlorine and the chloramine in that 1 gallon. You might notice that you get just the slightest tinge of brown color. So you might be able to detect that you have indeed put that humic acid into that water, and now it's safer for you to use it to dilute your soil sample. So we usually start at a 1 to 5 dilution, 1 gram or mil of your soil sample and 4 mils of the water. And now you want to shake that, because now we want to actually destroy all the structure that's inside. We want to break up those micro aggregates and the macro aggregates. And the way you do that, and we've done a lot of testing over the years, is if you think of your arm as going out straight, parallel to the ground, we want to go from here to a 90-degree angle and back again in 1 second. So I find it's very useful to have my watch give me the second. So they're noting by the second. So you go 1, 2, 3, 4. So each second I'm going from here to here, and you do that 30 times.
I'm also sitting here thinking that like a physical therapist would probably be able to say, pronate this muscle and rotate this other muscle, but that doesn't help most people either.
No, not at all, because I can get so confused and you end up being a pretzel and going, I'm not understanding this right.
Yeah, and it's not really hard. Like I don't want to scare anyone away. It's a very simple motion. It's just difficult to describe.
Yep. It's fairly simple, so shouldn't have any problems with it. So now you've shaken your sample, you've gotten all of those micro aggregates broken open. Anything that's not water stable and a little bit of water into the soil and shaking will pretty much break everything up. Now you want to wait probably about 10 seconds to let the bigger, chunkier stuff fall to the tip of your test tube. And if anybody needs information on where to buy these test tubes and with caps so you can shake, just get hold of us at info@soilfoodweb.com. And we'll give you a couple of choices of places to go with exactly the right kinds of test tubes and caps so that you don't have to be searching all over your town. Now back to our sample. Okay, so now we're going to take a pipette. And there's another thing that you want to buy is a pipette, and same sort of deal. You can get 1,000 of those for $12 or $15. And now you get your batch of 10 pipettes. So the first one, you want to put your pipette just below the top layer of water and then pull that sample into the pipette. Push it back out. We want to get all of the sides of the pipette where you inoculate all of the sides. So you're not losing any organisms to the fact that they're now adhered to the inside of your pipette. The second one, pull it up, push it out, pull it up. There you go. There's your sample. And you're going to put just one drop on that microscope slide. But you want to push that drop out very slowly and let it drop onto your microscope slide without touching the pipette tip to the slide. So it's got to be gravity is what's going to determine when that drop falls. And so a drop that will fall through the air, disjoint itself from that pipette tip, is going to be the same size no matter what size pipette you're using, if you do that correctly. So now we've got our microscope slide with a drop on the surface. We're going to take a cover slip and push the cover slip back and forth about the width of the cover slip, and then drop the cover slip onto the drop. You should have just enough water to cover the cover slip, no bubbles in it, no drops outside or inside. So now you're ready to put that microscope slide on the microscope and turn the lamp on. Make sure you focus in with the 4X lens. Go up to your 10, check your focus, go up to your 40X lens, and you should start seeing all the cute little critters. We do want to adjust the condenser. There'll be a knob on the left-hand side of your microscope underneath the stage, and you'll want to adjust that so that you're maximizing the amount of light. We can get really particular about getting the light just right, and that's probably an important thing. It's hard for me to demonstrate this to you or talk you through it. So that's one part where you would have to go to a class or something and get a little bit of training. We do have equipment lists on the website so that you can see all the equipment that you need to buy. It's a microscope with the right lenses. You've got to get a microscope with a condenser that's focusable, and it has an iris diaphragm.
And when we're looking now, you have it all focused, you have the lighting correct. What are we looking for? I assume that just seeing things may not be indicative of healthy soil. There's probably some specific indicators.
Yep. And we want to be looking for the bacteria and the fungi. The good guys are fairly innocuous. They're cock's-eye shaped round or a little bit longer than they are wide. The bacteria that are really scary are the ones that spin like a corkscrew. And you see that wave of motion as the corkscrew is zooming through your sample. And probably about 90% of the spirilla, that's their name, probably most of them are disease causer. And so if you see spirilla, you want to wash your hands. You want to make sure that you get rid of the sample, and wherever your soil is or whatever that sample was that you were testing, you're going to want to go out with a really good compost tea and just drench that soil with a good compost tea to kill the disease-causing organisms. Another one for bacteria are spirochetes, and they move like snakes, if you will, a wave motion. And the body usually has several of those S curves on them. So very noticeable, hard to mistake. When you see your first one, you'll go, yep, that's what that is. You want to be real careful because there's some really nasty diseases with those sets of microorganisms. Probably only 50% of the spirochetes are actual disease causers, but who wants to take a chance? And with both of those genera, they are indicators that your soil, your compost, whatever you're measuring, has gone anaerobic. So now you've got other problems that besides just the disease-causing organisms, you got some really nasty chemicals that have been made and drop the pH down to probably somewhere around 5.45 or 4, somewhere in that range. And putting that on your plants is not going to be beneficial and could actually kill them.
If you detect that in your compost, can you correct it, or you just need to get rid of it at that point?
You can correct it. It's going to take you a little bit of time. And certainly if you train with us, we go through all of the ways to prevent those anaerobic conditions from ever happening.
So there's indications along the way that kind of help you stay on track before you have to pull out the microscope and get to that level of
absolutely.
Yeah.
One of the best and easiest to figure out is when you walk up to your compost pile first thing in the day and you go, okay, who peed in the compost? Nope, your compost pile is going anaerobic. The smell of ammonia, the smell of vomit, the smell of sour milk, and then, of course, the rotten egg smell, any of those smells indicate to you that you better turn this pile and turn it pretty soon. Or maybe punch some cores down to the bottom. So a PVC pipe as tall as your compost pile is, and you're going to take that PVC pipe and pound a chimney into the compost. You pull the PVC pipe out, leaving a chimney where oxygen's going to get all the way down through your compost pile. And of course, hot air rises, cold air comes in, and so you're helping to drop the temperature on that pile as well. Depending on how hot you're getting, determines the number of places you have to put chimneys in. So we go through all of the criteria for turning your pile with you. What's good, what's bad, what is like, get the PVC pipe out here now. And once 9 o'clock at night, I walked out to my compost pile, checked the temperature, and it was at 175 degrees. And that's like you're 4 degrees shy of your pile becoming spontaneous combustion. It's going to burst into flames. And of course, this was in California in September when all these forest fires were going on, and it's like, great, I don't want to contribute to this problem. I really don't want to burn the house down, please. So I just took that PVC pipe and riddled it with holes all the way around, dropped the temperature about 3-4 degrees. And well, that was 9 o'clock at night, and I wanted to go back in and go to bed. I set the alarm every hour so I could go out and check the temperature on that pile. It wasn't going to turn it in the dark. And we had a bear and her cub in the vicinity. So I didn't want to stay out there nursing my pile. So come dawn, I'm out there and I turned that pile as soon as I could, but there were a few moments of don't ever put that much high nitrogen into a pile ever again.
I had no idea that composting could turn into such a dramatic event, like risk of spontaneous combustion with a mother bear nearby.
Yep. Yep. There was one time we put some fish hydrolysate into one of the compost piles to try to make it go a little faster. And during the night, Mama and Baby Bear came over and tore the pile apart. We found pieces of the wire cage, this, that, the thermometers just spread all over the place because they could smell that fish. It smelled like a fish was in there. So if you have a mama bear and her baby anywhere around, don't put fish into your compost pile.
Yeah, good advice.
The things we learn. The things we're looking at for fungi is we want them uniform diameter all the way along the body. If you get a branch and it's a different size, fine, not to worry. We want to know that it's a dark brown color, light tan. It's colored in some way. That's pretty much a signal of a good fungus. If it's clear, if it's colorless, a little bit more questionable. They should have septa inside of them, cross walls, and you can see the little cross walls. If you don't have cross walls, especially if it's clear, colorless, I would worry a fair amount that you don't really have the beneficial fungi that you want. Another factor works in here. If you see 5 strands of really good fungi and 1 short strand of bad guy, don't worry. The good guys are going to win. It's just when you have the only thing you've got are clear, white, very narrow diameter, no septa, you want to do something to get the good guys back into your pile. So under those conditions, you want to get a handful from another pile that you've got really good fungi in and inoculate it into the pile where you don't have any good guys. And it is that easy to bring them on.
So one thing I'm wondering about here is, people in general, and I'm a person too, are lazy. And I'm wondering, are there any proxies or indicators for soil health that don't require this level of commitment and investigation?
It's your nose. So the smells, if you're digging into the pile and you're starting to get bad odors out, you got to get it turned. If you're putting your hand into the pile and if you pull it up and you've got water streaming from your hand, it's going to be going anaerobic sometime very soon. So don't let the pile get that wet. If you put your hand in, pick up a handful of that compost, you want to squeeze it and you want just 1 or 2 drops of water coming out. That's perfect. You're not likely to go anaerobic if you've got the water right. You can pick up some of the pile, shake it, and you want to see lots of airways and passageways. Otherwise, chimneys. Get the chimneys going for you. And that's always a good way to assure aerobic conditions. So you don't have to suffer through, what do bad guy fungi look like?
And then for the soil itself, so composting is one thing. And for example, a friend of mine was telling me about how they moved into a new house like 25 years ago, and they weren't quite sure what they were getting. They wanted to have more native plants and make a better habitat. And they noticed just a huge number of slugs on the property. And slugs as detritivores are probably a little bit closer to soil than certain other organisms, and they're also observable. Was that an indicator of anything? Are there other sorts of visual indicators that might be helpful for a homeowner just trying to initially assess the state of their property?
Yep. If you are seeing a lot of snails, either slugs or snails, the slugs and the snails have come into that area because the soil is compacted. And if you can't get oxygen into your soil, the anaerobic organisms start making alcohol. And the slugs and the snails are attracted by that. And they'll have a perfectly wonderful time taking down the alcohol and then turning around and chewing on your plants. A little green, a little alcohol, a little more green, a little more alcohol. So that is really not good news to have the flood of the slugs and snails coming in. How do you fix it? You're going to want to go out with a broad fork and punch that into the ground and walk it back and forth. Go on, do it again. Just try to get a little fluff in the soil. And then as soon as possible, get out there with a compost. With the compost extract, you're just going to extract the biology from the compost. And then you use that. You don't need to put food into it and grow the organisms, because we don't have to be concerned about the organisms sticking to the surface of the soil. they're going to manage just fine to do that all by themselves. So as soon as you've got an area fluffed up with the broad fork, you put in that compost extract, and pretty rapidly you're going to be able to go deeper and deeper. One good test for that kind of thing is, where is the compaction, is if you take a metal rod and push it into the soil, and then you find out how far down is that anaerobic compaction layer. You can buy a penetrometer, which is the metal rod, and on top of the metal rod, there is where you can measure how many pounds per square inch are you pushing in order to push that metal rod through the soil. Most plants cannot push their root systems through a soil that's compacted any greater than 150 psi. You have plants that have the bottom root will go through, it can punch through and get deeper. So any of the things that, like the bulbs, can do that. So if you've got that special strength, they may be able to make it down to 300 psi. But I've never encountered a plant that can get through soil that is compacted at more than 300 psi. So you've got to get the biology back into that soil so they will build that structure for you. And I've seen organisms break up pressure areas that were up around 1200 psi in 4 weeks. They can do it rapidly, but you got to make sure that you've got the organisms, you've got the foods for them in that soil, you've got the mineral compounds that they need to break up to get structure built that rapidly.
So the first step of taking the fork and loosening the soil, that's to speed the process up and make it easier than when you apply your compost or the tea, as the case may be.
Yeah, because when the slugs are coming out, you don't know whether the alcohol is from way deep in the soil coming up as it's evaporating, or whether the compaction's right there at the surface. You don't know. Mm-hmm. And so the broad fork to get things started, and then it's a lot easier to start pushing that metal rod through the soil and trying to figure out where is the compaction.
And if a property has been treated with chemicals, and I expect there's going to be a wide range of timelines here. I maybe just start with fertilizers, with inorganic fertilizers. Is there any special steps that would need to be taken to flush that out of your land, or you just need to wait? What would you do in that situation?
Yeah, if you knew when the fertilizer was applied, you could have some idea of whether it's worth your effort to go get some really good compost right now and try to tie up all of those nutrients and keep them in your soil. It's just that you want to convert it from the salt form, the inorganic salt form of fertilizer, into your organic matter really quickly. Just getting a compost extract out into that soil is going to suck up those nutrients rather rapidly, so you keep them. It's a waste of money that they just, in the water going off, leaving your property, is taking all those soluble nutrients with them and causing problems in everybody's lakes and rivers and streams and the groundwater that's all negatively impacted by that inorganic fertilizer being lost. And it will be lost because fertilizer companies recognize over the years that they've had to increase the amount of inorganic fertilizer going on because there's nothing left in that soil to hold it. It is the biology that holds those nutrients.
That's really interesting to think about, in that you can start on this slippery slope by applying these fertilizers that then start destroying the biology of the soil, which then causes you to apply more. And then in the case that I was asking about, where now you're trying to restore an environment, it's not that you want to get rid of what was previously applied. You want to try to hold on to it and then restore the biology at the same time. And I guess I'm just repeating this because it will help me remember it. That was not how I thought that story was going to go.
Yeah, you thought you were going to get a lesson on how to leach all these horrible things out of your soil. Well, it's different if they applied something that has a lot of heavy metal in it. That's a different story because we don't want the heavy metals in the soil. So if you know that something that somebody else applied had a heavy metal in it, Now we definitely want to get the mycorrhizal fungi back into that soil, get them growing on the surfaces of the plants, because the mycorrhizal fungi will sequester the heavy metal just outside of the root system of your plant. There's been conjecture about why that works that way, and the best anyone can figure is that the plant can detect when that heavy metal is approaching, coming through the mycorrhizal fungal hyphae. And it will basically say to the mycorrhizal fungus, you aren't bringing that stuff in here. And it just stays in the fungal hyphae outside that root system. So it's tied up. It's going to, as the mycorrhizal fungi decompose, those heavy metals are going to be put into the structure of the organic matter in the soil. And as long as you're always increasing organic matter, the part of that organic matter that has the heavy metal in it will never be decomposed. It's not going to harm your plant because it's in the wrong form for your plant to take it up. Nobody else wants to decompose it because it's like, ew, what did I touch? Yeah, heavy metal. When you have somebody who's stupid and they dumped the diesel oil or they cleaned their engine out and all the gasoline went out on the property, you might over the years see some problems generated because of that. And when you start picking that up, then it's very easy to put in inocula of microorganisms that are specifically meant to decompose that stuff. The biggest problem usually then is you're not quite sure what it is that's causing that. It's like we've had ranches in sheep country where you just can't grow anything in this field. It's no matter what you do, you can't get anything to grow. And then you discover by talking to all the old guys in the community, you discover that this is the old sheep shearing pen. And this is where they had to put the animals into drenches of copper and a couple other real nasty things in order to get the fleas out, the ticks out, you name it, and you don't want them in your wool anyway. So arsenic, that's another one that they used quite often there. And so now you've got this whole section of this pasture, and now you understand why. Now that we know that it's arsenic, we can get the right mycorrhizal fungi out there to take up that arsenic. And again, it's outside the root system of your plant, so that you remove the plants, you take the whole root system, soil and all, and put it into a container where you can go and separate out the arsenic from everything else.
Before we started recording, I was telling you a bit about my audience and how there are a lot of people that probably self-identify as naturalists and are community science oriented, using iNaturalist, in fact, and other things. And I'm wondering, the topic of soil, it's something that I haven't seen a lot of interest in from that community. Do you have a call to action for those folks? Like, what would you like them to start reporting and documenting out in the field?
What would be useful is when they're expecting to see some plant in a community, the expectation is that whole community should be there. But there are some glaring absences. And is that because we don't have the right mycorrhizal fungi in the soil to allow those rare and endangered species or close to rare and endangered species? Can we go out and find the mycorrhizal fungi, extract some of those spores from the mushrooms that are produced by those mycorrhizal fungi, or from the roots? Small amounts of the roots could be harvested, and then you apply that to a land where this plant should be. Maybe get a few seeds going, put a couple of starts, and we could start to repair the lack of diversity that we have in some of our natural areas. I want to be careful about it's indigenous. We're not going to be going outside the area to collect the mycorrhizal fungi, the spores. There's a lot of the higher predators that they eat the organisms that eat the organisms that eat the protozoa, that eat the bacteria. And we're missing one of those larger predators somewhere along the way that's supposed to be present that would help make this natural area more typical of what it should be, get more production growing. Diversity is really important, helping any community fight diseases or pests or problems. So yeah, they need to understand their soil. It's not like the above ground goes on without stuff from below. Got to understand the soil if you want to have a healthy above ground system.
So yeah, that sounds like good advice for land managers as well. And then let's just talk about soilfoodweb, your organization. I know you offer classes that go really in depth into some of the topics that you touched on here today. Tell me a little bit about that and where people can go to learn more.
Great. Well, we have two companies. One is the soilfoodweb School, where we offer the foundation courses where we get pretty deep into the science. We teach people how to use the microscope at a fairly elementary level. And then the course after that is we go way deep into becoming a lab technician, running a lab where people send samples in. And then to become a consultant is the third stage. We think of it as this is your PhD in how to help people convert their dirt back into soil. So these consultants are trained in a number of environments. So they know how to help people, work with people. How do you make compost? How do you make teas and extracts? They can help you do that, where essentially they're getting people started on the understanding of what they need to have so they can become certified. So we have a number of advanced courses. There's always new stuff happening. There's new ways of measuring carbon sequestered in your soil, and we're considering an advanced class on doing that. How could we make this easier with microbes? Epifluorescent microscopy, some of that work where you can identify a lot of the pathogens by applying specific stains that are attached to something that reacts with salmonella or something that reacts with pasteurella, any one of those disease-causing organisms. And that's the only part of the sample that will start fluorescing. And you can see it in the microscope and know that this is real. This is attached to a bunch of the bacteria. So lots of different advanced classes going on that we're trying to increasing the number of advanced classes for people to come back to the school. We are planning on having annual meetings where people would be presenting. We do have the Soil Summit is coming up here pretty quick. It is all done virtually, so there's no travel to a convention area. We need to add that, I think. So another 4 years, 5 years, we'll be getting seriously into that. I've had a company called soilfoodweb Incorporated since 1996, and I've gone through several permutations with them. And so the soilfoodweb School is of soilfoodweb Incorporated. In the last couple of years, I've been considering what am I going to do with soilfoodweb Incorporated. And we've decided that's going to be the hands-on school, that people take the introductory, the foundation courses, and then once they're finished with those, now they can come and do hands-on making compost, extracts, teas, doing conversions of dirt to soil, because a lot of people don't have a place they can do these things. If you live in an apartment, where can you find space problem areas that need the dirt converted back into soil? So we're working with an 800-acre farm right now that needs to be complete. All 800 acres need to be converted. So that will be happening. We'll be working with grass seed growers. We'll be working with nut growers. We will be working with a number of large consortiums of people that everybody wants to be able to convert their dirt back into soil because it reduces cost by so much money. And then they can also get carbon credits for storing more carbon in the soil. We're working with some really large companies in Malaysia, Philippines, India, South Africa, Ecuador, Brazil, where people want to do this conversion. So If you want to wander around the world for the next 10 years and be paid for doing that, you might be interested in coming and learning to work with us on these projects. They are definitely large scale, and we're going to need literally thousands of people to do all the acreage that we're looking at already.
Sounds extremely interesting. And do you have any other resources that you would recommend for people if they're interested to learn more about soil health or the soil microbiome or even composting?
There's all kinds of books starting to come out on, soil food webs. There's a book by Jeff Lowenfels called Teeming with Microbes. And that's a really good starting manual for you because it goes through, it was published back in probably the late 1900s, 1990s. And things have progressed since then. But the basic knowledge in that book it still applies very well to today. You'd pick up most of the references in the foundation courses because everything that we talk about in the foundation courses have links back to scientific literature, back to USDA or NRCS literature that would get you right into the middle of everything. If you're interested in graduate school, you want to be looking at papers, the scientific papers, and who's doing what, finding a major professor that's doing a project that you want to do, or they're doing that kind of research.
Okay, thank you for that. And I know there's so much more we could have talked about. Like you touched on the fact that healthy soil is useful for carbon sequestration. We didn't really even get into that. There's so many more branches we could have gone down.
We didn't talk about the fact that you don't have weeds. If you're controlling the biology in your soil, weeds are a thing of the past. Protecting your soil surface, how do you do that? You put in ground cover. Well, what kind of ground cover? So there's lots of topics we haven't touched on yet. Yes. And that reminds me of another really good reference for people to go looking for. The USDA back in 2000, or maybe it's 2001, they published the soil biology primer. And out of I think it's like 8 chapters in that book, I wrote all except 2 of them. So Andy Maldinky wrote the chapter on microarthropods, and Clive Edwards wrote the chapter on earthworms. So it's a good place. And that has the picture of the soil food web in it. I think we only had 5 overarching principles. At that time, we're up to 7. So that's increased.
Is there anything else that you feel like is a burning topic that we should attempt to cover?
There's just so many burning topics. We've got to stop destroying that which supports us. And it's almost criminal to me that there are big chemical companies out there lying through their teeth that all of their toxic chemicals don't harm anything. Well, they use different laboratory techniques. Those techniques never will be able to isolate most of the microorganisms in the soil. It's like anytime you use a plate count, you're missing 99.99999% of the organisms in that soil. And so they use plate count methods to say, we have a sample of the soil without the toxic chemical, the sample with the toxic chemical, with 10 times higher concentration, and we look at them and there's no difference in the number of organisms that come up on any of those plates because you can't grow those things that are harmed by those toxic chemicals. And yet the USDA, the EPA lets them get away with that. It's just mind-boggling. So we've got to get ourselves out from under the corporate thumb. We'll get there sooner or later. So we need this study. We need to understand. We need everybody who possibly can to be putting this biology back into their soils so they're going to get all of the benefits of the nutrition, of no diseases, no pests, no toxic chemicals in the food that they're consuming or in their water. We got to do all of those things.
Dr. Elaine, it's been really fascinating, and I appreciate all of the information you've imparted here today. Thank you so much.
Well, thank you for inviting me. It's a good opportunity to reach more people. And that's what we have to do, is get everybody having a good biology in their soil. So whatever they're growing has all the benefits from the organisms in the soil.
Thanks for sticking through the entire episode. If you made it this far, I hope that it means that you enjoyed it. If so, please spread the word and share this episode with 3 friends or groups that you think would enjoy it too. As for today's episode, let me know. Did I miss anything? Was there a topic I should have covered? Let me know at podcast@jumpstartnature.com or DM me on any of my social accounts. I'll do my best to answer your questions. You can find me at Nature's Archive, one word, on Instagram, Facebook, and Twitter. I also share photography, nature stories, and much more on those accounts, so you can follow just to stay in touch, too. And despite being called crazy by numerous friends and colleagues, last year I left my tech career behind to start Jumpstart Nature, which Nature's Archive is now part of. For the sake of myself, my family, and the planet, I need to make this work. So please also consider becoming a patron at patreon.com/jumpstartnature. I offer some exclusive content and perks, and you can start donations as low as $4 a month. Lastly, please also check out our latest creation. It's the Jumpstart Nature podcast. We just completed our pilot season, where each episode reveals an unseen, surprising, or misunderstood nature topic with the help of experts and our host, Griff Griffith. It's entertaining and inspiring, and even reached number 3 on the Apple Nature podcast charts. There's much more on our roadmap, but we need your support, so check out jumpstartnature.com for more details. Thank you.